Well, hello and welcome everyone. I'm Libby Mucciaroni from the Organic Trade Association and the Organic Center. I'm so pleased to welcome all of you to our virtual field day, Integrating Livestock and its impacts on food safety and soil health. The Organic Center is thrilled to present today's webinar along with our research partners. As I discuss a few of the housekeeping items, please acquaint yourself with the speakers that are here today. We're so thrilled to have so many people that have registered and such a really dynamic, fantastic group of speakers today. So a couple of those housekeeping items that I'll share with you now before we get to the actual show. First of all, the webinar is being recorded today. Any resources that we reference throughout the webinar will be shared both in the chat box, and then we'll send a follow-up email afterward that will include all of the information that you possibly want to know, as well as a follow-up survey, which will also appear after the webinar ends in your browser. During registration, we ask that you let our presenters know any questions that you might have on today's topic. You can use the Q&A box in Zoom to let us know whatever questions you may have. We'll be monitoring that throughout, and we will have two different Q&A sections today during our hour-and-a-half webinar to get all of the information that you want to get understood about today's topic from our speakers. They are ready and excited to talk about this research and all of the work that they've put into this. The other thing I will quickly say is that there's going to be, a really great dynamic set of presentations. Please use that Q&A as frequently as possible. The more questions, the better for a dynamic Q&A. And finally, if you happen to have any questions throughout the webinar today, please feel free to, technical questions rather, please feel free to use the chat and message me directly, or you can also use that Q&A box as well. So before I hand things off to the main speakers, the reason why you're here today is we're letting folks continue to jump in. A couple of things I'd like to let you know about. First of all, the Organic Center is a nonprofit research and education organization based out of Washington, D.C. They work to conduct and convene credible evidence-based science on the environmental and health effects of organic food and farming, and to communicate the findings to the public. The Organic Center's work is supported by companies and individuals who desire a sustainable and secure food system that promotes the health of humans and the environment. The Organic Trade Association is a membership-based organization for the organic industry. We ensure that all parts of the organic value chain have a strong voice in government and in our communities. We bring farmers, processors, distributors, retailers, and others together to promote and protect the growing organic sector. OTA represents its members to government on sector needs, market development and promotion, and strong organic standards and regulations. Our members receive the latest information and quick answers on organic regulations and standards in the United States and around the world. If your organization is not a member of the Organic Trade Association but would like to know more information, please let us know in the chat and we will get you in touch with our membership team. And with that, I'd like to hand things over to Dr. Amber Sligo, the Director of Science Programs at the Organic Center. She'll be here to talk a bit about today's presentation, our research partners, and the presenters today. Thanks so much for being here.
Thanks, Libby. I can't tell if you can see me yet, but I think my video is on. So anyways, hi, everyone. I'm excited to see you today. We haven't had an Organic Center webinar in a while, it feels like. And sorry, I can see the videos coming off and on here. I just want to make sure that we're okay. Libby, does everything look okay on your end before I continue?
Yeah, things look good.
Okay, great, thanks. So yeah, we seem to be following a bit of a trend in our past webinars, today's webinar, and then a future webinar that we have coming up shortly too. And that theme is food safety. And this has been a topic that's had a lot of popularity in the past, and I think it continues to get a lot of attention because food safety regulations, they're remaining diligent, and organic farmers are also continuing to search for this balance between meeting those food safety regulations, satisfying organic standards, continuously improving their environmental impacts of their farms as well as their bottom line. But before I dive too deeply in today's topic, because I don't want to give it all away, I'd like to start the run of show today with just a little bit of background on the Organic Center and how we got to the work that we will be presenting today. So for those of you who are not super familiar with the Organic Center and what we do here, I'll just sort of reiterate some of Libby's introduction there and say that we are a nonprofit organization. We were founded in 2002 with a mission to conduct, convene, and communicate science that's related to organic food and fiber production. We do work very closely with the Organic Trade Association. They help us connect to a vast network of farmers, farmer organizations, industry members, food brands, who we then are able to connect with academic researchers so that we can collaboratively develop these research projects that will address organic farming needs. What this process can generally look like is that through our networking, we identify knowledge gaps, and then we help pull together research teams and help raise funding to conduct the research that will fill those knowledge gaps. And that's basically how we started this collaboration with today's presenters. Next slide. So a lot of this work began about 8 or maybe 9 years ago now, when the FDA proposed revisions to the Food Safety Modernization Act. If I or others refer to this again in the future, we'll probably say FSMA because it's easier. But those revisions included changes to the required wait period between the application of untreated manure and the harvest of the treated crop. The FDA at the time had proposed a 9-month minimum interval requirement, and this directly contradicted the National Organic Program regulations for the application of raw manure. Ultimately, that ruling was not adopted because the FDA decided that further research was needed. And with that recommendation, TOC reached out to Dr. Alda Pierce and other academics across the country, many of whom are on today's webinar, to help develop research that was aimed at making sure that the organic systems were included in that collection of additional data that the FDA would then consider for future rulemaking. Some of that research was presented across 2 webinars that we hosted last year. And Libby, at some point, if she gets a break from all of her technical stuff on the back end, can maybe drop a link to those webinars for any of you who are interested in looking at the on-demand recordings. And the topics for those webinars were how to use animal-based soil amendments to improve soil health while also considering and minimizing food safety risks. And the second webinar was on science-based composting practices that growers can use to minimize food safety risks. And today's webinar is an extension of all of that work. And I'll say too that something that's kind of special about this research team that I don't always see is that, you know, while they started with this one topic in mind, which in this case was measuring the potential food safety risks of raw manure, they have continued to work together and continue to raise funding to research additional topics that have all sort of evolved and spawned from that original work. So today we'll be discussing food safety impacts of cover crop grazing and fresh produce systems with a focus on cover crop adoption, crop livestock integration, and soil health. Next slide. So I am grateful to be joined today. Oops, I think I'm missing my notes here, sorry. This is not what I wanted to say to you all. Here we go, sorry. We go back, Libby, we changed the order of these slides too many times, I guess. So there we go, perfect. So we know that planting winter cover crops and integrating livestock into cropping systems can have important benefits for growers as well as the environment. But these practices don't come without some challenges as well. So for instance, cover crops, we know, can enhance soil fertility. It can improve soil structure, water infiltration, and storage, which is super important for droughts and flooding. And they can also reduce nitrate leaching. But There are some challenges in expanding the use of cover crops, partly due to concerns over cover crop water usage and nutrient immobilization, which can lead to a decrease in basically nutrient bioavailability and ultimately input costs for the following crop. Many growers also do consider livestock grazing of cover crop fields and in fresh produce operations even as a way to enhance soil health and environmental benefits. It can increase carbon inputs and nutrient cycling. However, there have been concerns about microbial food safety, and this can be limiting the expansion of this practice because livestock grazing can introduce fecal-borne, food-borne pathogens into soil, and there is a potential for transfer from the soil to harvested produce that will be consumed raw or minimally processed by consumers. Next slide. So the goal of this project is to evaluate the food safety and soil health impacts of ruminant grazing of cover crops by measuring several things, including food pathogen persistence and survival in the soil, as well as transfer to vegetable crops, changes in soil health indicators over 2 years of grazed cover crop vegetable production, and the benefits and the potential trade-offs of crop productivity. This experiment has been conducted across 3 states with different climate conditions and growing seasons to see whether these benefits and challenges are regionally dependent. Next slide. This project is led by the University of California, Davis in partnership with the Organic Center, as well as USDA's Agricultural Research Service, ARS, the University of Maryland, Eastern Shore, and the University of Minnesota. Funding for this work, this particular project, was made possible by the US Department of Agriculture's Agricultural Marketing Service. I'm so used to using the acronyms, USDA's AMS. Next slide. All right. So I am grateful to be joined today by an esteemed group of presenters. I've been working with this team for many, many years. So I'm excited that we all can be together here today. From the University of California, Davis, we are joined by Dr. Alda Pierce, Dr. Amelie Godon, and PhD students Sejin Chong and Sequoia Williams. And from the University of Minnesota, we are joined by Emily Evans. We will start today with some background on the methods of this work, really highlighting the differences in regional requirements. And then we'll show you some videos and photos from the field that will demonstrate those regional differences. That's sort of the field day component of today's presentation. And then at that point, we'll stop and open it up for any questions that you might have on the methods at that stage. Then we'll move into some preliminary results presentation from this work, and then we'll land on our final Q&A. So I think that's enough talking from me. And I'd like to hand it over to the first of our presenters, Alda, to give you a little bit more detail about this research project.
Thank you, Amber, for a very nice introduction and background of this multi-year collaboration. I've been working with the Organic Center for a couple of years now in different aspects on food safety of use of enriched raw manure and biological cell amendments. So for today, I would like just start with overview with our definition of what are diversified small-scale farms and integrated systems. The audience might be less familiar with this term, and sometimes we see in the literature use different terms meaning the same. So diversified and integrated crop livestock systems are farms where animals and crops are raised with the goal of utilizing products of one for the growth of the other. There are many advantages of this integration of animals, and we see you can see that integration in 3 different ways or classified in different ways. Sometimes you see these animals in the pen, so they are separate from the growing areas. of the crops and in pens, or we see in the middle what we call rotational. Some of these farms, they have a semi-permanent pastures about 2 years or more, where they move animals depending of the pasture for 2 or 3 years, and then they switch that pasture for a crop growing vegetables or other crops. Or down here, they may use grazing animals as sheep for their cover crops. That's the topic of today, or residue crops. So here down we have a sheep grazing on sweet corn after that corn was harvested for marketing. Another system may come across what we call full combine. So they have the animals on top, we have broilers. They move these pens every few days along of orchards, or use sheep for vineyards, where Dr. Emily Godin will share some of her work she has done on these systems. So this kind of different integration of crop livestock systems, as mentioned before, they use some of these inputs of the animals, namely the manure or untreated biological soil amendments. And we know that manure can carry foodborne pathogens or pathogens with public health importance. Some of these animals, they tend to be reservoirs for these pathogens. So it's normal to find Salmonella, E. coli on 5.7 in some of these animals. For example, ruminants is known for E. coli on 5.7. So when we apply this manure on fields where we're going to grow vegetables that normally eat raw, there is a food safety implications or risks because they have not been treated to reduce those pathogens. So some of the recommendations might be, let's apply to those untreated manure to crops that's not produced normal. They have a killing process, for example, potatoes. Although what do the farmers, they do, that is a way to reduce that risk through applying a time interval between application and harvest. And that's what our past work has been done in collaboration with this team, looking at that time intervals on application of untreated manure or applying a treatment to the soil amendment. The treatment to soil amendment is not possible for using grazing animals. So what farmers are doing as they move livestock from pasture to pasture or to produce area, either can use on crop residues or cover crops, they will follow time interval as well. So here on the side, you can see some of these rotational grazing systems. You might see these side by side. So we have these on top, chicken coops, they move the chicken coop side by side of some of these vegetable crops, or they use the sheep for the cover crops, winter cover crops here in California, or the crops. So the manure is deposited while grazing, so there will be sources of fertility and organic matter. As I mentioned, this is untreated manure, so there is a potential for source of pathogens as manure application. So what this implies planning from the farmer, how they plan these rotations, how they plan this growing season, and quite a planning to avoid that cross-contamination when they use animals for grazing. So what growers are following, or most of these growers are following, is the National Organic Program of 19-120 days standards or rule. And this is the recommendations for application of raw manure. Although we know that there is less manure deposit when you use grazing animals because there is a timing that the animals are moving from paddocks to paddocks. So should be this treated as wildlife intrusion as event? So am I thinking as a wildlife intrusion? Although for farmers, it will be easier having a time interval that they can apply. And that's actually what they are doing. So with that in mind, we did an on-farm project. That's how we start actually this rotational grazing multi-state. Was a farmer, was it using these rotational grazing cover crops. I was very interested to assess those risks. So this paper was, or this work was published in 2018, and was one farm here in California, and where to evaluate the persistence of E. coli in soil on these diversified farms. They were integrated sheep on vegetable crops. So in this case, it was in collaboration with the Western Center for Food Safety, Dr. Michelle J. Russell. In this case, we didn't have experimental design. we just observed what farmer was doing. So I'm going to just give overview of some of that data. So what we did, we follow that soil that we have two fields, field A and field B. Field A here is in yellow, and field A is in blue. So we sample repeatedly this soil and test for foodborne pathogens and generic E. coli. Why use generic E. coli? We use as a fecal contamination indicator because foodborne pathogens is relatively rare. So just to explain a little bit of this graph, on the y-axis, we have generic E. coli concentration on log, and x-axis the timing post-ship. If you look at on the yellow graph, so field B, we start sampling the soil after the sheep left, and we see a peak on increase of that concentration of generic E. coli right after the planting and change of irrigation system. While field A, we start following up later on, and there is kind of already, there's after the peak, both fields we see a die-off or a decrease of that concentration. So what are really interesting is these 19 and 20 days, what happened there? And we saw that about 84 to 100 days after grazing, the concentration of generic E. coli drops for one log. Oops, there's something happened here out of my control. So we saw that drops. Let's see if I can go back. Here we go. One more. Okay, so sorry about that. So maybe I've clicked too much on my mouse. So after we saw there's a drop of the overall generic E. coli. So this shows how important is monitoring the farm for management practice. So you might say, yes, generic E. coli is environmental, is in the soil. How we can, is that generic coli related we find in the soil to the sheep? So what we did PFG that time, that's what we used to looking at these isolates that we collect from fecal samples for that flock and the soil. And what we found was what we call 4 physi-soil clusters, meaning there's 4 clusters that are very identical to generic E. coli. And for example, here highlighting in yellow at 84 days post-shipping was identical generic E. coli found in the soil and the fecal samples. So what do we know in background, we know from other studies, we know that the shedding of these foodborne pathogens, Salmonella, pathogen E. coli in livestock, depends on the species. Here we're using sheep and goat for this study. So depends on the species and also the health status. In the past, we found an average of prevalence of STEC in sheep flocks between 3–30%. Also depends on diet and some other stress-related events, for example, transportation. Some of these animals are used for grazing. They might be rented and they may move from farm to farm, as well from the grazing density or stock density. In the soil, there are many different factors as environmental factors, climate, so weather-related factors, soil soil characteristics, soil type, microbial population, and so on, and irrigation, as well the cover crop. So the next question is how we can study these factors, how they interplay in survival of these pathogens in the soil. So with that in mind, that's how we come up with this study across 3 states. So looking at difference or not, that we're going to observe through the different regions. And we're looking at the effect of cover crop systems in sheep grazing, in persistence of foodborne pathogens, indicators of contamination in California, Minnesota, and Maryland. And then Emily group looking at also the nutrient cycling, soil health, and on vegetable crop and across these 3 regions. And finally, the third objective was provide information, outreach program and information on field and online, targeting producers and other stakeholders interest in these systems. So as a final slide for today, this section I have, so as I mentioned, we did 3 states here in California at the Russell Ranch. So they are research farms, part of the UC Davis. In Minnesota also at the research farm in the Southwest Research and Outreach Center, and Maryland in research and teaching farm at University of Maryland Eastern Shore. So with that, I will end my presentation for now, and there will be Dr. Emily Godin talk a little bit of background of this project as well.
Hi everyone, pleasure to be here today. I've been tasked with sharing a 5-minute introduction to soil health and nutrient cycling in grazed annual cropping system. And I wanted to start with this concept that land-based integration and really reconnection of livestock and crop and forest production really has the potential to improve multiple dimensions of environmental and soil health, while providing avenues to positively impact animal and human health in this, if these systems are really optimized to maximize some of the beneficial ecological functions that are stemming from livestock grazing on organic cropland. And so recognizing really that the health of animal, of people, and the environments are connected, this complex agroecosystem, which involve multiple actors, can really help reach the goal put forward by various institutions to enhance the role of agriculture to reach One Health through collaborative efforts between farmers, sheep producers, and veterinary and soil health communities. And we've certainly learned a lot from farmers who have been implementing some of those practices. So we find various examples of integrated crop livestock system around the world. These systems remain the backbone of subsistence agriculture. In industrialized countries such as the US, ag system, including organic production, are increasingly specialized and decoupled with crop and animal production, largely contributing to poor nutrient recycling within and between agricultural operations. And the current model for integrated crop livestock system really, as you can see in this picture, employ very diverse, adaptable, feasible management practices that can be very strategically implemented across various crop production systems to facilitate a transition toward some of those more soil-centric, internally regulated management paradigm. And that helps create a circular or semi-circular flow of nutrients and energy, as well as better using ecosystem services that are provided by animals for organic production. In California, where those examples are that you see here are from, we sense really a regain of interest by organic growers for this practice. And here you can see some example of grazing alfalfa forage or grazing of cover crops in orchard and vineyards. Now, adoption in organic vegetable system is still low, mainly hampered by the very little data we have on long-term outcome for productivity, soil health, and of course concerns about food safety post-grazing. So really, by impacting the amount and the quality and the timing of organic material that are entering the soil, grazing of cover crop ahead of vegetable production can have a very, very large impact on soil ecosystem, and we need to better understand the impacts if we want to move forward. So soils are this very diverse and heterogeneous ecosystem. They support a large biodiversity, and that biodiversity is really cornerstone into regulating nutrient cycling, water resource conservation, as well as the soil physical and chemical property relevant for crop production. And so their capacity to support food production while helping with climate change adaptation and mitigation is really encompassed in this beautiful metaphor of soil health. And that really brings forward the far-reaching potential of soil ecosystem to build sustainability, but also resilience of our agroecosystem. So there's many definitions of soil health. What does that? One of the most common is recognizing that soils have a continued capacity to function as the vital living ecosystem to sustain plant, animal, and human. And soil health is defined by various factors. And to make an analogy, you know, our health is a function of our gene and genome that we receive from our parents, the environment we live in, and our actions such as diet and exercise. While soil health is similarly a function of the soil parental material, the environment, temperature, precipitation, vegetation, topography, and the action that growers take through management decision. And it's often measured using a suite of indicators that represent the physical, chemical, and biological dimension of soil. But when considering the ecological impact of grazing on soil, it might be useful to kind of recenter soils not only as a suite of disjointed physical, chemical, and biological parameter with specific target, but really considering it as a living ecosystem with inherent to each region. Where these three types of indicator really are intrinsically connected. So we have the physical characteristics that regulate the soil physical habitat for flow of water, air, and nutrients, physical protection and retention of nutrient resources, including carbon, and the diversity and regulating the diversity and activity of soil organisms. And then there's another complex set of interacting resources which serve as energy sources for soil organism and for supporting plant production that is especially important in organic systems. So as such, really a healthy soil should increase cycling and plant availability of nutrients when they need it, should store nutrients during fallow of period of low plant uptake, and should provide an appropriate physical environments for resource exchange and conservation. So grazing really can impact multiple components of the soil ecosystem. Now we have an increasing amount of information on cover crop. Cover crop is cornerstone to a lot of very successful organic production systems. And although much more data is needed to better understand their impact in specific location, we know that cover crop can provide nutrients through the composition of biomass, energy, and habitat for soil community to do their job, help having adequate soil structures through those root channels, for instance, and channel for water flow, protect soil from erosion, and help break pest and disease cycle, including weeds, which are really a tough management challenge in organic production system. Now, when you start grazing, then you can see here, I put two pictures. On the left is a grazed residue, and on the right is a mowed and chipped residue. And you can see that grazing can really drastically shift the amount and quality of residue left on the field. Integration of animal changes the soil physical habitat structure through varying degrees of physical disturbance via trampling and what we call hole-faction that incorporates the dung, the urine, and the residue and modify those physical properties of soil. As you can see in these pictures, it might be more heterogeneous cover crop termination, and the effectiveness and impact will vary according to stocking density, timing of grazing, but also animals and plant species. Grazing also impacts soil chemistry, and in particular the nutrient content through processing of residue that enter the soil ecosystem and that are providing often more rapidly bioavailable nutrients which are more biologically active. I wanted to show you this diagram that shows a little bit more details what happened in the soil, and especially nutrient cycling when a grazing occur. First, the plant uptake nutrients from the soil matrix or the atmosphere and couple it with carbon obtained through photosynthesis into biomass. The animal processes and use carbon and nitrogen in their body to change the chemistry and form of the residue. And so a cover crop with a mix of legumes and cereal that has a carbon to nitrogen ratio of 20:1, for instance, is going to be processed into dung and urine, which have a lower carbon to nitrogen ratio. Now, in addition, plants and animals are amazing because they've co-evolved for millions of years to maintain ecosystem health. And so plants have developed amazing mechanism to recover post-grazing, which can actually benefit agricultural production. So as you can see here, grazing often triggers a reallocation of plant resources below ground. So we see an increase in root growth, especially in cereals, more root turnover and exudation of organic carbon compounds that we call exudates. And those compounds are really cool because they're preferentially used as a source of energy by soil microbial community to do their job. We can also see flushes and hotspots of inorganic nutrients that might be available for leaching, but really the manure and urine entering the soil provide a balanced and more diverse and biochar source of nutrients that is really close to the microbial needs and are therefore thought to be more readily incorporated into microbial biomass. And so that really allows efficient nutrient recycling and storage. And we think that this practice is actually extremely synergistic with compost application and cover crop to make really better synchronize nutrient availability with crop needs. Now, this organic matter also allows storage in non-leachable form, and that can be mineralized back into bioavailable nitrogen. And why really nitrogen export occur with the animal, we've calculated that it's less than 20% of nutrient export. Grazing often increase microbial biomass and activity and diversified nitrogen input, so you can have this staggered nutrient delivery to the crop. Uncertain, I think, remains impact on compaction and productivity, but we're really starting to, especially in annual system, but I wanted to show you here some nuggets of data that we have in perennial system, where we've been working with farmers in our landscape that had 10 plus years of integration of livestock. The first project is an almond system and led by Krista Marshall, a master's student in my lab, where she's monitored 12 different almond orchards, which are on similar soil type and texture, have provided a management gradient and looked at different soil health properties across those different orchards. And those orchards are clustered. The cluster A's are orchard with bare soil and just organic inputs such as compost. The cluster B has winter cover crop and some organic inputs. And the cluster C is where you can start it, where we have animal integration and animal grazing. And on those normalized parameters here, you can really see that animal integration, as seen on those green lines, improve most of the soil health parameters that we measured in terms of soil organic carbon, microbial biomass, labile carbon, total nitrogen, P and K, fungal biomass, and indicators of soil food web activity. When we look at the multifunctionality, so the capacity of our soil to provide multiple function important for organic producers, we see that animal integration really increased significantly the ability of our soils to function and to provide benefits for ag production. We've also looked at this in vineyards that were organic or not in the Napa Valley, grazed with high density, short duration sheep. And where are my slides? Do you see the slides?
Hi, I think Libby just lost her connection and I believe she was sharing the slides. So I'm going to go ahead and pull them up. If you can just give me a minute.
Yeah, and so in vineyards, really what we see are similar increase in soil organic carbon across soil depths, more nitrogen and phosphorus availability, especially in the surface soil. We see a larger soil microbial community that is more active, and we measured no differences across perennial system in terms of soil structure. So no increase in bulk density, no increase in compaction. And I realize that that comes, with difference in grazing practices, but it's not something ubiquitous that we find across the system. So I think in summary, these provide some benefits, especially to organic producers. It helps provide nutrients and biological activity into the soils, help processing of residues that are sometimes hard to manage, reconnecting nutrient cycle, and also adding dollar value to cover crops, and helping to reach some of those regenerative ag market. We also see some other cool benefits in terms of weed control, and we're measuring this, but we see a shift in weed community and potentially lower weed pressure after long-term grazing. So results may vary tremendously. Growers have reached out with really interesting questions regarding goat versus sheep, different type of cover crops, and we have so much more to do. But results will vary according to disturbance level, timing and stocking density, the type of animal, and the knowledge and experience of growers. So with that, thank you. I'm not sure what's going on with the slides. I'm not in control, but I have concluded my presentation.
Thanks, Amelie. I think
Thanks.
Naomi's been able to recover for Libby here in the background. So we can just go ahead and move on with Alda.
Thank you, Emily, and thank you, Amber. So this next session, I'll just give an overview before the videos. How does look like these field trials across 3 regions? As I mentioned, we are presenting some of the data from the year 1. We repeat these experiments 2 years, so we are finished up the 2nd year. So we are doing the same study design here in California, Minnesota, and Maryland. In part, we see different climates. So in blue, it will be California in different graphs, semi-arid. Maryland has a humid coastal, and Minnesota humid continental. How does look? So the study design or the experiment trials were done on a research farm. We conduct a randomized complete block experiment. They look all the same across the 3 states. We have 3 treatments, and you'll hear often through this presentation. We have the fallow, so no cover crop. So winter cover crop tilled, the cover crop is tilled, and winter cover crop grazed with sheep or goats. We repeat this 4 blocks, and here is representing those blocks with different treatments. And we have 1 plot or subplot that's grazing each block. So how does this look like across the states? And that's what here represents some of the image across the 3 states. We use the same type of winter cover crops mix with cereal, rye, crimson clover, and radish mix. When that cover crop is mature, actually they look quite different. Here that's just on the left in California, just see on the picture below the sheep arriving to the cover crop, and then on Minnesota in the middle, and Maryland. So the cover crops look quite different. Also, they have grown different seasons. In the California side, they use sheep, about 40 ewes. We have a farm at UC Davis, so we use the sheep from that farm. In Minnesota, they rent goats, and they have actually 2 grazing seasons, grazing events, one in the fall with 40 goats, and another in the spring with another 15 goats. And Maryland, we use sheep per grazing plot. So 2 places or 2 sites with sheep and 1 with goats. So the next slide is give overview how looks like the weather or different temperatures across the different states, and what implications in terms of field work does have. So in the first slide is the monthly average temperature across from August to June, 20 to 21. In orange is Minnesota, and green Maryland, blue Davis. And we have cover crops was seeded at different times. So August was in Minnesota, here in California in November, and Maryland was the following year that was seeded that mixed cover crop. In terms of precipitation, also cumulative precipitation is totally different across 3 states as well. Maryland did have 14 inch of rain with snow, and Minnesota less rain with 28 inch total with snow as well. California going through a drought for a couple of years, so not much rain, which limit actually us to doing just one grazing event for our spring grazing. So next few slides, just kind of comparative with the image before the videos. We're focused on California and Minnesota for this comparison. So while in August, Minnesota already have established cover crops, California was preparing the field. Then in November, that's when we did our cover crop seeding here in California. There's the picture on top. And Minnesota did have their first grazing event with goats. So they were able to do 2 grazing events between snow. So California just had 1 grazing in February, and part of this was we didn't have much rain, so we limit how much we grow that we were able to graze. And Minnesota at the same time, they were burying snow. So we have Emily and Lee here doing some soil sampling after a snowfall. Then moving to the spring, we already have by April established spinach in our field tries here on top in California, and Minnesota did have their second grazing. They actually have a surprise kidding it with the triplets, and goats just have triplets, you see their kids. So they were able to do 2 grazing events, one in summer and another one back in spring after the winter. So the harvesting of spinach. So the first year we used spinach, second year we used cucumber. So this is the findings of the first year. So harvesting of spinach was between May and June. So May earlier on here in California, and then June later on for Minnesota. One challenge for us to use spinach. So we used 2 different produce, one with contact, one was spinach, and another is cucumbers. The challenge for us, where I locate our research farm in Central Valley, we have very short timing for that spinach before it gets really hot. That's the overview of the timeline between the 2 states. And next we'll have the video with some of those field work and logistics on the grazing for the 2 states as well. So I think Libby can go and or the organic center. So we need some time to start the videos, to switch to the videos. Okay.
Great, thanks so much. You can turn off the video. We're going to do a brief Q&A. Again, everyone, I apologize for some of the technical issues that we've had today. Thanks for your patience. There's been so much really fantastic conversation already from Alda and Amelie's presentation. Naomi, if you have a moment, you can actually spotlight everyone so that all of our speakers are on. That would be really fantastic. And while I am doing this brief intro, I'll again encourage folks to keep sending questions. We've had over 25. So we're going to cover a few right now over the next 7 to 10 minutes, and then we're going to move on to some additional presentations and then have a real large 20-minute, hopefully, opportunity to discuss so many of your questions, as many as possible. So the first question I'm going to ask is a little bit of a level setter here. And this was submitted that we got from one of our pre-registrants. So my question is multi-regional research on the ground from a research coordination perspective. What are some of the biggest challenges that you faced, and how has the pandemic exacerbated this? Who wants to take a stab?
I think I'll answer that question. So we have, as Amber mentioned, we did collaborate with the same team since 2000, for quite a few years in another project. And they have a great team, but I think one of the biggest challenge is with pandemic or not pandemic, we have very, very different growing seasons, right? So as you could see from my slides, events happen different because there's different growing season, nothing to do about it. What that implies are actually our data comparable as a researcher, how I can compare these 3 states while we have more than 120 days after the last grazing for Minnesota and the second grazing. So that's one challenge for us, comparing and standardize those data points. Another challenge we had last 2 years was the pandemic. So different institutions have a different policies regarding COVID. That was a big challenge for us to start at the same point. We have a great team of colleagues and PhD students. They were great coordinating all this work. Definitely did have a layer of complexity in terms of logistics. But bottom line, I think, is as a researcher, is growing seasons, different management of the animals and so on, how we come up with either a timeline or recommendations that will across states.
Great, thanks, Alda. Does anyone else have anything they want to jump in there on about that, or about their experiences? Hearing no one else jumping at it. So here's another question. So based on your perspective from conducting this research, what do you see as logistic challenges for farmers to integrate livestock and food safety aside?
I can answer that. We recently had a focus group in California that we brought together farmers and ranchers and kind of had that discussion. And so after talking to them, we realized that there is, especially when grazing is seen as like distinct operations, like not necessarily one diversified farm with both crops and livestock, but rather like a farmer with land that is used for crops and then a rancher with livestock, and bringing those 2 systems together. The timing of farming operations can be really hard for the growers. Growers don't want to delay planting their crops. It's like another thing to think about is getting animals on their land. So that can be a logistical issue. And then for ranchers, they have to keep their livestock healthy and alive, which farmers sometimes don't always think about because they just deal with plants. So animals are a little bit trickier sometimes, as we saw with some getting born on our fields and stuff like that. And in general, it just requires some sort of trial period for farmers and ranchers to try out this practice, maybe on like a smaller sized field, and kind of it also requires just a shift in systems thinking and thinking about what these the landscape as a whole and integrating livestock into the landscape as a whole. So
great, that's really interesting. Anyone else want to say anything about that?
I can say something from the upper Midwest perspective.
Great.
We have such a short season that a lot of times it's limited by finding shelter because of the harsh winter conditions, or alternative food sources because they simply can't graze probably more than 60% of the year. So that's limiting. And it seems to work out that a lot of organic farmers, crop farmers included, they tend to have neighbors that have enough manure that they're willing to give it away. And so they still find a way to integrate livestock without actually being solely responsible for the livestock.
Great, really thank you so much. So I have one more question I think I'm going to ask now, and then I'll move us on to some more presentations in the interest of time, and then we'll hit all the rest of the questions, as many as we can, at the end of this session. So my question here is, do you think that ultimately for the progress of organic agriculture, that livestock has to be integrated into cropping?
I can take this question.
Great.
So it's a challenging practice, and I mean Sequoia has explained very well, and Emily, you know, some of the pitfalls there and complexities. I think it's definitely the next level of ecological intensification. I've never seen soil like this in about 20 years of looking at soils in different systems. Once you start integrating an animal, you really shift your soil ecosystem. So from a soil health perspective, it's really puzzling the large impact that it can have, and the potential that it has to bring your ecosystem to a different state. So it's definitely a next level of diversification. It helps reconnecting different enterprises in the landscape with large potential. We see it being very effective in other parts of the world that we can learn from, especially in combination with no-till. Definitely an increasing pillar of regenerative agriculture. So regenerative agricultural model have the principles of soil health building plus animal integration and no pesticides, so no biocides. So definitely the next step forward, in my opinion, for diversification and intensification of organic systems.
Great, thank you so much. Well, I know that there are dozens of questions to cover, and I think we could just stop here and keep going for the next hour, but I'm going to pause us now. Thank all of you for your responses thus far, and I'm going to hand things over to Sequoia Williams in just a moment. I'm going to set everything up here, and we will keep going with the presentations, keep asking your questions, everyone, and looking forward to even more conversation in just the next few moments here. Thanks so much, everyone.
Cool. Thank you, Libby. So I'm going to present a little bit about our results in soil health and yield from our first year in California. And I'll try to make this a quick presentation so we can get back to questions. Let's see. Okay, so kind of going back to Amelie's presentation about soil health, I have a definition here that says soil health is the degree to which dynamic properties have been managed for optimum function within the constraints of the soil's inherent properties. And so in this presentation, I'm breaking down these dynamic properties into kind of the traditional physical, chemical, and biological components of soil health. I have a table here that shows some of the various metrics that are measured in assessing soil health, and the ones that I'm going to be presenting on today are circled in yellow. So I have some data on bulk density, which is a measure of soil compaction. I measured wet aggregate stability, which correlates with water infiltration rates and can impact erosion or lack of erosion. Potentially mineralizable nitrogen is a metric that falls under the biological category because it has to do with microbes mineralizing nitrogen through the cropping season for the plants to uptake. And active carbon, which is another microbial biological metric that gets at the carbon availability for microbes. And then finally, in the chemical column, I'll just be presenting on two, which are kind of the ones that probably farmers think about a lot, which is nitrate and pH. And then the last part of this definition is the fact that this is all happening within the constraints of a soil's inherent properties. So I just came from a soil field course at Davis in California, where we looked at a ton of different soils across the state. And essentially, I just wanted to share a reminder that different soils can have different limits to their potential to build carbon and things like that. So for example, in this picture, the soil in the middle can potentially store more carbon than the soil on the left. And so for our 3 states that we're looking at, we have 3 different soil orders that we're looking at. And this is something that will limit the potential for each state to build certain aspects of their soil health. So now getting into some of the results. This is results from 2020 to 2021. And I was looking at samples that were taken kind of during the peak of the spinach growth, essentially. And we were sent all of our samples for all 3 states to the same laboratory in Maine so that we had uniformity with our analyses. So the first one I'll talk about is aggregation. Aggregation, like I said, allows for greater water infiltration if there's good aggregation, and also provides habitat for microbial activity. So for our first year in Davis, we didn't see any differences. So this graph shows the fallow plots in orange, the ungrazed cover crop plots in green, and the grazed cover crop plots in blue. And that'll be the same for the rest of the graphs as well. So you can see that there was no significant changes to water stable aggregates between our treatments. And it's likely that because this is an annual tilled system, that tillage is probably a greater control on aggregation rather than grazing. And then the next thing that we looked at was bulk density, which, like I said, is a measure of soil compaction. And we also didn't see any increases in compaction, which is really promising with grazing. And the other thing to note too here is that this field site in California has actually had grazing this was the 3rd year that we had grazing, although it was only the 1st for this project. So this is actually kind of looking at more of a slightly longer term effect than might be expected, or for the other states at least. Now looking at the soil health chemical properties. Like I said, I looked at pH, and we didn't see any significant changes to pH with grazing. It was the same across all 3 treatments, essentially. And the next one we looked at, oops, I missed the last one. Here we go. Soil nitrate. So this is where we're starting to see some shifts in the soil when it comes to nitrogen, which is to be expected with grazing. Like Amelie was saying, grazing kind of processes a lot of that cover crop material and inputs it as manure and urine, which then can mineralize more easily than the cover crop biomass itself. And so we saw significantly greater soil nitrate at spinach planting in the ungrazed cover crop and the grazed cover crop, with slight trends towards more in the grazed, but that wasn't significantly different than the ungrazed. Finally, the last part of the soil health triad is the biological. And so we looked at respirable carbon, and we saw some trends towards greater respirable carbon with grazing, although it wasn't statistically significantly greater than the other 2 treatments. And we also looked at potentially mineralizable nitrogen, which had trends towards greater mineralization in the grazed plots as well. And then, of course, I'm sure farmers want to know, what about the yield? So we looked at spinach yield at our trial, and we saw, significantly greater yield in the ungrazed cover crop and the grazed cover crop. And again, we saw trends towards higher yields in the spinach plot, in the grazed plots, although it wasn't statistically significantly greater than the ungrazed crops. So this is promising, although, yeah, it's we'll have to look at kind of all 3 states for the 2 years to kind of get a better picture for all of these metrics. This is just some of the preliminary data that we saw in California for the 1st year. Yep, so in summary, for soil health in California, the soil physical health indicators were not impacted by grazing. That was the bulk density and the aggregation. We saw some shifts in some of the soil chemical health indicators, notably with nitrogen and greater nitrogen availability. And we also saw some trends towards greater microbial activity in the grazed plots, with our potentially mineralizable nitrogen being higher and trends towards greater respirable carbon. And essentially, as we move forward with this data analysis, I just want to remind you that we have like 3 different states with 3 different soil types with differing potentials to build soil health. So we'll keep that in mind as we do our data analysis going forward. And that is all I have for you all. So I think I'm going to hand it off to Sejin now.
Thank you, Sequoia. Hello, this is Sejin Chang, a second-year PhD student at UC Davis working for this organic integrated crop livestock farming project in Dr. Aldo Perez lab. Today, I'm going to present some year 1 data of foodborne pathogens we've tested for this project. So the objective of our study was to evaluating the prevalence and persistence of generic E. coli, E. coli 0157, non-0157 Zika toxin producing E. coli, and Listeria in California and Minnesota field respectively. So here, generic E. coli is tested as an indicator of fecal contamination. So we quantify the concentration of generic E. coli in soil samples. And for the other 3 pathogens, since they're actually foodborne pathogens that could cause illness to human, we tested for the prevalence. For the study design, the field had 4 replicates of 3 treatment types. The 3 treatments were fallow, winter cover crop, tilted without grazing, winter cover crop grazing by sheep or goats. So the fallow part has no winter cover crop and no grazing events. And in this figure, the green background plots indicate where the winter cover crops were seeded. And the area inside of the red line indicates where the grazing was done. So red line depicts the electric fences we installed. And here SL stands for soil sample numbers. So we sample 36 soils in each sampling day, which was 12 samples per treatment. And this is the timeline of how we sample soil, feces, spinach. So soil samples were collected every month, including baseline and day 7 post-grazing until day 120 after the grazing event, to see how long the contamination of pathogens persisting in the field after the grazing event. And we also collected 20 fecal samples pre and post-grazing event, and 24 of spinach samples, which was our year 1 produce, were collected as well. There were 3 differences between California and Minnesota field. First, California used sheep as a grazing animal, but Minnesota used goats. And second, California had only 1 grazing event, but Minnesota had 2 grazing events according to their weather and cover crop growth. And third, California harvested spinach twice, but Minnesota harvested once. As a result, in soil and spinach samples, no foodborne pathogens isolated in both California and Minnesota field, but in fecal samples, some positive non-0157 SPEC were observed in both California and Minnesota field. And in Minnesota, Listeria was also detected in some of the pre-grazed fecal samples. Based on this result, this means even though there were some positive foodborne pathogens detected in fecal samples, there were no transfer of those pathogens to soil or spinach in year 1. And for generic E. coli, which was tested as a proxy of fecal contamination, here graph shows changes in concentration of generic E. coli in soil among 3 different treatment groups over time. The y-axis of this graph shows the log transformed concentration of generic E. coli in soil, and x-axis shows the sampling day with 3 different treatments. The 3 different colors of box plots depict the 3 different treatments. So here the gray color was fallow, the blue was tilted without grazing part, and orange was the grazing part. And in both California and Minnesota field, the mean concentration of generic E. coli were highest at day 30 post-grazing only in the grazing area. And in other fallow and tilted without grazing area, the concentration of generic E. coli were constantly low over time. Also, when comparing 3 groups in each sampling day, a significant difference among treatments was observed at day 30 post-grazing in California and day 7, 30, and 60 post-grazing in Minnesota. Also in Minnesota, there was no even positive generic E. coli in all treatment groups in day 90 and day 120 after the grazing. So in conclusion, data from our year 1 show minimal risk of transferring foodborne pathogens to produce after 90 days after the grazing. And additionally, we're still working on assessing the effect of environmental factors such as soil composition and weather and microbial population on presence of generic E. coli. This is all I have, and I'll pass this over to Amber.
Great, thanks so much, Sejin. I'll welcome everyone to come on camera, all of our speakers, as we move on to our Q&A here in just a second. I'll leave this up for a second. Keep asking your questions. We've had so many fantastic questions. Hopefully you've seen responses coming through. Really excited to dig in more in the next 15 minutes collectively about so many different diverse questions that we've had coming through here. So my first question, as you all get settled in, grab a drink, get ready to speak a bunch again collectively, is a question that Amber actually posed to all of you. So you're all familiar with the question, ready for it to come through. She asked the question, how do you anticipate climate changes to impact food safety risks? I know you haven't processed all of the data, but do you have any sense, based on your past work with raw manure, what to expect to see with livestock integration?
While you all are thinking about this, I'll expand it and just say that, we just landed on this point by Sejin about environmental factors. And I know you all are still processing that, but just thinking about, as we have, if you're in a place where you end up having hotter temperatures or colder temperatures or more rain or less rain, what are some of the most important environmental factors that you expect would impact, the food safety risks in these systems?
So I'll try to answer some. I don't think we have answer for all these questions. We don't know, but that's why I keep studying these areas. We know that change, so we can see different ways. One is what this drastic change on weather does have on animal itself. What we know is some species, they shed more foodborne pathogens in certain seasons or under certain conditions. An example, we know that cattle share more salmonella across the studies during the summer, or STEC. So what are the effects does have this change in animals itself that lead us to shedding more foodborne pathogens and potential cross-contamination of soil? That's something to consider. On soil itself and food safety, we know some of these pathogens, they can regrow, so they kind of dormant in the soil, and they can re-multiply, I will say, and for example, after rain events. So it's not uncommon to see an increase of these pathogens in the soil. So how are we going to minimize these risks? It's all about be aware of the risk and what we will change in our practice. Are we, and it's a risk assessment based approach. And that's part of why these on farm and on, real life situation does help try to connect all these dots and observe in nature. And it becomes very complicated, right? So what you can change to minimize that risk. So we have to think about those parts. So integration of animals, what does do on animal itself, and how that survival pathogens might increase or not with periods of extreme events.
Great. Thank you so much, Alda. So we got a question that actually was related to the video that I want to throw out there because I think it's an interesting question. And this is from Evie. She said that she saw a goat rental truck in the video and it got her thinking about management. Is the idea that these types of systems would require a farmer to hire goats specifically to graze the cover crops once or a few times a year? Or could a farmer theoretically manage both goats and a crop for profit? So like another overarching question that she posed out there is, what is the ideal management system that you're thinking about with all of this research? Hopefully there's a thought or two percolating in these brilliant minds here with me. And if not, we can move on and we can answer that later over email if that's helpful.
I can speak a little bit about the goat rental. So at our research center, we didn't have livestock. So that's why we went in that direction. But that goat rental is something that farmers around here, this might not totally relate to your question, but they use it for taking care of buckthorn, taking care of thistle patches. It's another organic means to control weeds. So it's just an option that we have here. So I think sometimes when we're talking about an ideal system, it depends very independently on what your goals are. And sometimes these farmers just need very specific quick fixes and they can't support animals on their farm. I don't know if that's ideal, but that's just the kind of reality of what is happening in our area right now.
So just having that in California, we have actually livestock owners that they will rent their animals, sheep, farm to farm, to go and that's one of the challenge for those who want to start adopt this practice, because there's a high demand during spring and so on. So there is actually a website that starts with collaboration with the Cooperative Extension in Sonoma, is Match Graze, where they match owners with livestock and people that would like to have their property graze. Here with the fires, we have used a lot of start using much more small ruminants for fire suppression and decrease biomass and potential in these different rural areas, suburban areas. So that is that business model here already in place.
I actually really, sorry. I really like this question because it to me, it points out a couple different things. One, it shows how sometimes we're able to do things for research that aren't necessarily, they don't totally reflect what happens on the farm in real life. And two, I think this is a larger question about the future of integrating livestock, the implementation of this practice in our food system. What will be feasible in different areas for different people. And I think this ties into also thinking about the different types of animals. Like for this particular study, you've been working with sheep and goats. And maybe in some places, ruminant animals are not the next logical step for a farmer for integrating. And there are other regions where something that might be easier could be poultry. And I saw that listed also in one of the questions. And so I'm going to throw out the question to you all, how do you think these different animals would impact your results or the logistics? And I know that you've done some work in the past looking at different types of animals, but I just love to hear your thoughts on something, other than the sheep and goats. We just saw something about alpacas, a question about alpacas. So have fun with that.
That is a recurring question that I cannot answer. So there we go.
Great. So I'm going to pivot us over to a couple of questions that we received here. One was from Thomas. He said that Ariana von Bruggen, Kate Sko, and others have shown that human pathogens are less persistent in manure and compost-treated soils. Is your research confirming that? And what can you share about that?
So I will answer this question and try to tie it up to another question come up here on the chat. So Kate Sko group, they're looking at in vitro, so data from 20 years on the research plots where they compare in vitro inoculation of some foodborne pathogens of salmonella of those plots, meaning on if I'm not on bench. So they're looking at how the survival of salmonella and some other foodborne pathogens compare plots with compost and manure. And what they saw a suppression of those pathogens survive less on those plots that have been application of manure and compost. So Suji show the data from 1 year, but actually we've been looking at this system of grazing in cover crops and in food safety aspect for about 4 years. In 4 years of data here in California, we have identified only once, that means 4 field trials, only once foodborne pathogens in the soil. Although we found on fecal samples on the sheep, Aztec, Shiga toxin-like E. coli in the soil, we found twice now. And that was, one was not actually even grazed, was a fallow, and another was grazed 7 days after the grazing event. We're saying that consistently, foodborne pathogens in the soil is rare with the systems we are using, with this stock density, with this cover crop, and so on. Is that comparable in other systems we have to look at? Another question I had was, we harvest the spinach before 90 days and 120 days. So this study was done in certified organic farms, because that's what, some of the farms, they are using certified organics. And the certified organic farmers, they follow the 90 and 120 days. What that means, they pull out the sheep from the field 90 days before, if it's a vegetable that is not in contact with the soil. So they need to plan when they're going to plant their vegetables, or 120 days if it's a vegetable that's in contact with the soil, as for example, the spinach or leafy greens, right? Very close. So to avoid the contamination. These standards of 90 and 120 days were based on production cycle, not based on food safety. That's why we're looking at this project. If you look at and pay attention, and I see that people pay attention, so our spinach harvest was at 60, 90 days. And the reason was because spinach has a short growing season. We are in Central Valley, so we could not delay that planting. So it was a planning in our side. But we still looking at the soil through those 90, 120 days. So sampling, even though we are not harvest the spinach. That spinach did not go to the human consumption actually, because of the 90 and 120 days. So in summary, we have data for a couple of years. The trend and what we saw, that graph of generic E. coli. So because foodborne pathogens are rare, and we are not infecting, sheep with foodborne pathogens and try to have that high concentration in soil. We use in sheep that naturally infected. We use generic E. coli as a proxy. And that graph, that's a peak after 30, 60 days of generic E. coli, and then kind of dies off. So the other studies, we didn't see any difference after 90 and 120 days. So I hope that these answer some of the questions we have regarding foodborne pathogens. Another one was what kind of, just a detail, what kind of analysis or lab we use. Actually, I published that. I can share the protocols for enrichment and so on, confirmation of foodborne pathogens. Just to finalize, what interests to know, what is change of diet of sheep going, we use sheep from the sheep barn. They're using mostly dry forage, alfalfa, hay. Maybe what does constant change in their diet in my effect on not shedding? Actually, we're looking at, that's why we sample before and after, looking at how change on their microbiome, fecal microbiome, is there a big change after these grazing events or not? And relate that to the presence of pathogens. So a lot of questions, a lot of things we don't know yet, to try to understand better all these placed together. I hope so. I'll let another question, and I hope. Oh, there's another one regarding the certified inspectors. How we can get by have animals with certain crops. I think what I hear from farmers, and depends, right? There's some buyers, depends on the crop, and the inspectors, it might be okay. if you respect 90 and 120 days with your grazing animals, and maybe in another groups they decide they rather don't see any animals on the property. So it seems to me, I don't have data supported, but talk with farmers, it seems to me it's variable depending which group and commodity we're working with.
Great, thanks, Alda. I know that we have about 2 minutes left. I'm going to ask one at least additional question here. And then to those of you that we didn't get to answer questions, I know there's so many here left that we'd love to be able to engage, and we just kind of hit the clock. We will follow up over email. Our researchers are super excited to share more information with you. I have a question from Emily. She said, if a food safety team is resistant to allow any type of livestock into organic fields for grazing, what would be the number one thing that you would tell them to assuage their concerns? And I think as many of you as possible that want to answer that. And if we go up a little bit on time, I think that's fine. But maybe have asked the wrong question to wrap things up. It sounds like a little bit here. Maybe I'll reframe the question slightly from each of you. If you have like one key takeaway that you'd like to share to make sure the audience walked away from what you spoke about today or what we've discussed in totality, that would be a great piece of information to share out and have captured in the recording today. And maybe Alda, I'll have you start off and we'll just kind of work our way down. I'll call on you one at a time.
I think in terms of food safety goes back about knowing your risks, right? So knowing what is your flock, which animal species are you using, and what is coming. So I start one of the slides, animal health. No, have that relationship if you don't own the livestock with the owner, the rancher, and know the flock and the risks. Another thing that's, the recommendation we still have is the 90 and 120 days between the grazing, pull out the animals, and then harvest whatever your commodity. As saying that depends on the commodity. So if you're talking about, so here is a system where we till in the soil, we move the soil, and they might change the microbial population. So they might help to reduce the foodborne pathogens. No tilling soils, we don't have much work on that to advise one way or another way.
What I would say from a soil health perspective is that animal integration really shifts your soil ecosystem. It makes it more biologically active. There's more nutrient cycling, more carbon sequestration potential, which is kind of amazing to see in California, where it's hard to achieve some of those targets. Although it depends on the type of animal and the grazing practices, and it's still some questions that we still have a lot of work to do to answer some of those questions. I think having a strong relationship with the shepherds and the animal handler, and working this relationship and building trust and land-based knowledge is really important to realize this potential.
Great, thanks so much, Emily. Emily, any thoughts?
I think the one thought that it wasn't brought up, but it was one part of the Q&A was talking about management of the cover crop post-grazing and how that would either promote or limit the spread of pathogens. And I think that's an awesome question. And that would be really interesting to look into. I think that would have a lot to do with it.
Great, thanks so much. Sejan?
Thank you for this webinar, because this is year one preliminary data. I'm looking for like whole data set after finishing all this 2-year project from multi-state areas. Thank you.
Yeah, thanks, Sejan. I think it's a really interesting point what you're saying, that there's still so much to gather, and folks were asking even about when is this coming out? There's so much excitement. So I can't wait to see the publication. I'm sure all of you can't wait to see the work and the publication of all the results that you're finding from this. And Emily, to your point, it's it's so true that there's just so much research opportunities, and we're gathering them even in the Q&A. So we're only beginning to probably scratch surfaces, right, of where we're able to go moving forward in the future. I'll move over to Sequoia, and then Amber, I'll give you the last thoughts after that, if that sounds good. And I will also share a link in the chat before we go to that about how to sign up for other educational opportunities, and a quick note again that we will be sharing out the recording, edited, and all of the resources that have been talked about and answers to questions over email on the next day. Thanks so much.
Thanks, Libby. Yeah, I don't think I can add much to what's been said, but I'm really looking forward to diving into the soil health data from all 3 states for both years and seeing if we can find any trends.
Great. So I'll do the final thoughts, I guess, and say that I'm really excited about this research. It's clear that there are so many more directions that it could go. There's a lot of context dependency, but at least this is a great start. And I feel like this can provide some hope for people who are, wanting to lean more in that direction of figuring out how to integrate livestock more into our current food production system. I like that we're seeing less risk than what was originally feared, food safety wise. I like that we're seeing results that are showing, clear distinctions and improvements in soil health. So yeah, I just feel very excited about this work. Also, we keep talking about this being preliminary results. We are planning, the Organic Center is planning on hosting another webinar at the end of this study. So we will have the final results fully synthesized. That'll probably be another year, but stay tuned because we will have more to say about this.
Great, Amber, thank you so much. Again, I want to offer extended thanks and appreciation to each of these researchers. They put so much time and effort both into this presentation and just into all of the work that's been presented today. Thanks to our audience for being here and joining us. And we're looking forward to continuing the conversation, not only now over email, but also make sure to share this out with your networks. And we'll see you as soon as the research is ready to be presented again in a year. Thanks so much for everything, everyone. Bye.